Welcome to this tutorial on the internal anatomy of the brainstem. We indeed turn again to one of the most complicated parts of the nervous system, which brings to mind our first core concept in the field of neuroscience, and that is that the brain is the body's most complex organ. I have one learning objective for you today, and that's to be able to identify the major subdivisions of the brainstem as seen in representative transverse cross-sections. So in order to achieve this objective, you're going to have to be familiar with what we discover when we look inside the brainstem in each of the three major divisions, the midbrain, the pons, and the medulla. So here's the key in achieving this objective. I want you to mobilize your mental image of the external features of the brainstem as we begin to look at our transverse cross-sections. And if you can do so, you'll know what to expect to find in each of the subdivisions. So when we think about the external features, think about the form of each division of the brainstem. Think about the bulges, the furrows, the grooves, the columns that we might see from the outside. And think about the cranial nerves and how they attach. That will be key in order to interpret what we see on the inside. It all begins with our mental image. So that's what we're working towards, and in order to get there, I'd like for us to have a tour of the brainstem using Sylvius 4 software. So if you'd like, go ahead and open your version of Sylvius 4 if you have it. If you don't, that's fine. You can follow along with me. Okay, I've just opened up Sylvius 4, and I'm going to now click on the Brainstem Cross-Sectional Atlas, All Structures, and this will open up now our Brainstem Atlas. And this is an atlas of transverse sections through the brainstem, where the sections are prepared in order to resemble a standard histological myelin stain, which means white matter structures will appear dark as if they've been stained with silver salts, and gray matter structures will be relatively unstained, so they will be light. So the convention is essentially opposite what the terms usually convey. So white matter is dark and gray matter is light. And in Sylvius, we have a window to the left that shows us our navigational views of the content, including a list of thumbnails, some slider tools that will help us navigate along, and we can select an image to enlarge, and as we mouse over features within the image, we notice that a label appears, and if we were to click on that structure, it becomes highlighted in the thumbnails, as we can see over to the left, and a statement of structure or function appears in the text box to the right. So that's just a little bit about navigating in Sylvius. So let's go ahead and begin. We begin at the top of this atlas, or the most superior section, which is very near the junction of the thalamus and the midbrain. We recognize elements of the thalamus here, all this gray matter along the dorsal and medial aspect just to the lateral wall of the third ventricle. Down below we have the mammillary bodies, which is part of the hypothalamus. So we are definitely in a portion of the posterior diencephalon, but we are also starting to get into the midbrain, and we know that because of these massive bundles of white matter that are beginning to take shape in the ventrolateral surface of the brain. That's where we expect to find the cerebral peduncles. In fact, what we have here is the confluence of the internal capsule into the cerebral peduncles. So if I select the cerebral peduncles and then just go down one section, we are clearly in the midbrain, and the cerebral peduncles are one of those key features that will allow you to be able to recognize this part of the brain. Another feature would be what's going on with the ventricular system. So we just saw the third ventricle opening up in the region of the diencephalon. In the midbrain, we don't expect to see the third ventricle, rather we expect to see the narrow cerebral aqueduct. And indeed, that's what we see here. So the presence of the peduncles, the presence of the cerebral aqueduct is confirmation that we are looking at the midbrain. Well, let's think about, again, that outside to inside perspective. What else do we see from the outside when we think about the midbrain? Well, you should be thinking about a cranial nerve 3. And indeed, we have cranial nerve 3 here, the ocular motor nerve. And so, again, another characteristic feature of the midbrain. Cranial nerve 3 is derived from a somatic motor nucleus along the dorsal midline of the tegmentum of the brainstem. This is the ocular motor nucleus. It also has a parasympathetic motor component from the Ettinger-Westphal nucleus. So those are the cranial nerve nuclei associated with this level of the midbrain. But there's lots of other features here that's worth noting in the midbrain. There's a very large spherical nucleus in the midbrain that looks a little bit orangish in a fresh specimen of the human brain that has been sectioned open. This is called the red nucleus, very prominent nucleus of the tegmentum in the midbrain. Between the red nucleus and the cerebral peduncle is the substantia nigra, which is really two nuclei in one. There's a compact layer of cells on the dorsal margin of the nigra, and these are the famous dopamine neurons of the midbrain that project broadly to the forebrain, including the basal ganglia, parts of the prefrontal cortex of the frontal lobe, as well as the amygdala and the hippocampus. Then there is an inferior part to the substantia nigra, which is very much like the globus pallidus in the basal ganglia. We'll say much more about all of this when we talk about the basal ganglia. But for now, I would highlight that the substantia nigra is found in the midbrain. Okay, let's look at the dorsal part of the midbrain. And again, if we think about what we see from the outside when we look at the midbrain, you may recall that there are these 4 bumps that we see from the outside. They're called little hills or colliculi, which means little hill. There are a pair of superior colliculi and a pair of inferior colliculi. We are at the level of the superior colliculus, the superior of these little hills, one on either side of the midline. And just below the superior colliculi, around the cerebral aqueduct, is a bit of gray matter that we call the periaqueductal gray. All right, well, let's go down a section to a lower level of the midbrain. And at this level, we are below the superior colliculus, and we are at the level where we are beginning to develop the inferior colliculus. We're actually a little bit below that little hill strategically, because we want to see the nucleus of the trochlear nerve, which is found here along the dorsal midline of the tegmentum, right in line but inferior to where we saw the ocular motor nuclei. We still have a small cerebral aqueduct surrounded by a large periaqueductal gray nucleus. So that is consistent with what we saw in a more superior level. But now you'll note that the inferior part of the section looks quite a bit different. We still see something that resembles the cerebral peduncle, but now we're beginning to see some defasciculation of these axons in the ventral part of this section. In fact, what we're getting into is a set of nuclei that is defining the features of the pons. So this is very near the junction of the pons and the midbrain. We have nuclei in the base of the pons called the pontine nuclei that give rise to axons called the pontocerebellar fibers, because they're going from the pons to the cerebellum. Well, let's go ahead and section down into the pons. We'll actually skip this section and go to the widest part of the pons, where the pons is sending axons out into the cerebellum. And then we'll work our way back up to those sections we just passed by. Well, we're in the pons. How do we know that? Well, because we see these pontine nuclei defining much of this basal or ventral aspect of the pons, giving rise to this white matter called the pontocerebellar fibers. This accounts for that transverse orientation of axons we see in the pons that is bridging the long axis of the brainstem, pons means bridge, and these pontocerebellar fibers give rise to these massive bundles of axons that enter the cerebellum called the middle cerebellar peduncle. Now in the region of the tegmentum of the pons, we have some cranial nerve nuclei of note. We have the chief sensory nucleus of the trigeminal complex, also called the principal sensory nucleus, and the trigeminal motor nucleus that are connected up with the trigeminal nerve roots, which we see here. Now, above the tegmentum, of course, that cerebral aqueduct has now opened up into a large fourth ventricle. And so lateral and dorsal to that fourth ventricle is the cerebellum. Now, you may be wondering about these dark bundles of white matter that we see here in the lateral aspect of the ventricles here. This is the superior cerebellar peduncle, yet another major bundle of axons associated with the cerebellum. So we had the middle peduncle, by far the largest of the three cerebellar peduncles, and the superior cerebellar peduncle. I'm going to select this structure and then retrace our steps and go up a few sections and see what happens to this peduncle. This peduncle is the means by which the cerebellum sends signals out of the cerebellum to reach our motor circuits in the thalamus. And so these peduncles are leaving the cerebellum, and as they do so, they work their way into the tegmentum of the pons where they cross the midline. So we see a crossing of this peduncle called a decussation in the upper part of the pons. And that's really the primary feature I wanted to highlight in the sections between the level of the trochlear nucleus and the trigeminal nerve. So let's go back to the section that we were just viewing and highlight a few other features. We notice in this basal region, while many of the axons running through have defasciculated, we're beginning to see a recondensation of axons into a tight bundle. This will emerge on the other side of the pons as an important pathway called the medullary pyramid, which contains, among other axons, the cortical spinal tract. So we'll say much about that pathway in this course. All right, so let's go down one section, which is the level of the caudal part of the pons. And I'll deselect the superior peduncle here for a moment and show you some other features that are present in the tegmentum of the pons, again starting with cranial nerve nuclei. So now we have a different division of our trigeminal nuclear complex. This is called the spinal nucleus of the trigeminal complex. This is part of the trigeminal nucleus that extends from the level of the principal or chief sensory nucleus all the way down to the merging of the caudal medulla with the upper cervical spinal cord. So there's a nucleus, and then just lateral to the nucleus is a tract called the spinal trigeminal tract. We have other important cranial nerve nuclei here. We have the facial motor nucleus in the caudal pons, which gives rise to axons that sweep across the tegmentum and loop around an important gray matter structure just below the pointer here, and these axons then arc back out and exit the ventrolateral aspect of the pons. What they loop around is the abducens nucleus. So here's the abducens nucleus, a somatic motor nucleus along the dorsal midline of the tegmentum. It gives rise to the axons of the abducens nerve that exit near the junction of the pons and the medulla. We've accounted for the abducens nerve as it exits near the midline of the pons. We've accounted for the facial nerve that exits a bit more laterally. The third of these three pairs of nerves at this junctional region is the vestibulocochlear nerve, and at this level we're beginning to get into some of the nuclei that are associated with that nerve. This is one of the vestibular nuclei that we have in this region of the pons. Here's another one. Because there are multiple nuclei here receiving incoming vestibular signals, we call this the vestibular nuclear complex. All right, let's move now out of the pons and into the upper part of the medulla. Here, again, we expect to find vestibular nuclei. We have yet an additional vestibular nucleus here, part of that nuclear complex. But we also have the auditory component of the eighth nerve, which, because we didn't achieve an optimal section through it in Sylvius, we elected not to label it. But we see a little bit of it here. It's this gray matter that forms this rind around the outside of the dorsolateral aspect of the medulla. And what it's surrounding is a large mass of white matter that I'll get to in a second. So this gray matter that we see out here, this is part of the cochlear nuclear complex. So there are multiple divisions of this complex. We can call them collectively the cochlear nuclei. So we've got our vestibular nuclei and then our cochlear nuclei associated with the eighth nerve. Well, this big massive bundle of white matter, this is the inferior cerebellar peduncle, one of the three major stalks that attaches the cerebellum to the brainstem. This is inferior and it attaches from below, mainly conveying signals from the spinal cord, but also signals from an important nucleus of the medulla that I'll mention in just a moment. Well, we also have elements of the trigeminal system here. Here's our spinal trigeminal tract and our spinal trigeminal nucleus. We'll see this all the way through the medulla. Now, in the more ventral aspects of the medulla, those fibers that were penetrating through the basal region of the pons and coalescing back into a nice compact bundle of white matter have now formed the medullary pyramids. And at this level, these axons are almost entirely the cortical spinal tract. That is, the pathway by which the cortex governs the motor systems of the spinal cord. Just above the medullary pyramids is really quite a lovely nucleus. It's called the inferior olivary nucleus. And this is the nucleus that gives rise to a bulge in the ventrolateral aspect of the medulla that we call the olive. So you may recall from our surface views of the medulla seeing the columns of the medullary pyramid, and just lateral to it is the inferior olive, or the olive for short. That's what we call it from the outside. When we're looking at a cross-section as we are here, we recognize this as the inferior olivary nucleus. Well, let's move on. Now, you may remember between the olive and the medullary pyramid exits the axons of the 12th nerve, the hypoglossal nerve. We can see the hypoglossal nerve roots here as they pass between the pyramid and the olive. And their source is the hypoglossal nucleus, another somatic motor nucleus that we find on the dorsal midline of the tegmentum in the medulla. Now, just lateral to that nucleus, we have the dorsal motor nucleus of vagus. And right next to that nucleus is a sensory nucleus, the nucleus of the solitary tract. And that's gray matter that surrounds this solitary compact bundle of white matter called the solitary tract. The nucleus of the solitary tract is a visceral sensory nucleus. The dorsal motor nucleus of vagus is a parasympathetic visceral motor outflow nucleus. So notice again the organization that we have here: somatic motor, visceral motor, visceral sensory, and then we have special sensory and general sensory. Our special sensory at this level are still vestibular nuclei, and then our general sensory is still the spinal trigeminal nucleus, the spinal trigeminal tract. Now, I also should point out one important white matter pathway that we'll spend a fair amount of time on when we talk about our mechanosensory systems, and it is a medial ribbon of white matter that sits right on top of the medullary pyramids. It's called the medial lemniscus. So I'm going to select the medial lemniscus so I can show you how this structure, this medial ribbon of white matter, runs all the way through the brainstem. I haven't highlighted it until this section because we probably see it best in the middle to lower part of the medulla. But indeed, we can track it all the way up from the caudal medulla to the thalamus, and we will do so when we study our mechanosensory pathways. For this structure conveys our sense of touch from the body below the face, from the brainstem to the thalamus. Okay, let's go now to the caudal part of the medulla and highlight a few structures of interest. Now, what we find is still a spinal trigeminal nucleus and tract, but now in the dorsal part of this section, we've lost the fourth ventricle entirely, and what we have are a set of gray matter nuclei that are bounded by white matter pathways. And these nuclei are called the dorsal column nuclei because they sit right on top of the dorsal columns of white matter that rise up from the spinal cord. And there are two of them. There's a medial tract called the gracile tract, which ends in a gracile nucleus. And then there's a more lateral tract called the cuneate tract that ends in the cuneate nucleus. Now these nuclei, these dorsal column nuclei, they grow the axons that form the medial lemniscus. So they grow axons that sweep around through the tegmentum of the brainstem called the internal arcuate fibers, and they form the medial lemniscus. The medial lemniscus axons derive from the contralateral or the opposite side dorsal column nuclei. So there's a crossing, there's a decussation here. There's one other important decussation to note at this level of the medulla, and it's a decussation of the medullary pyramids. Notice how one pyramid is starting to stream up and over the midline on top of the other pyramid. If we go down one final section, we'll see this crossing more obviously. This is where the corticospinal tract crosses the midline. Actually, as we'll see in a few weeks, about 90 to 95% of the axons cross. Some remain on the same side, but the vast majority of them cross, and here's where they do so, in the caudal medulla. Well, that completes our brief tour of the internal anatomy of the brainstem. We'll have the remainder of the course to reinforce this anatomy and come back to it. We'll do so when we talk about our long pathways for somatic sensation and motor control. And then we'll apply this knowledge to consider the localization of injury in clinical cases where there's been some focal damage. And what will be important to emphasize at that point in your learning is the regional anatomy. That is, where do various systems come together in small places in the nervous system? And the brainstem is one such important place where even very small injuries of nerve tissue can give rise to widespread and disparate neurological signs and symptoms. So I look forward to having that level of discussion with you in a few weeks, but for now, take your time and go through the brainstem sections and identify the principal gray matter and white matter structures that are highlighted for you in the tutorial notes, including those that I've highlighted here in this video. And use this knowledge. We'll apply this, we'll discuss it as we go along. So thanks for your attention, and I'll see you next time. Welcome to today's tutorial on the cranial nerve nuclei. This topic pertains to one of our foundational core concepts in the field of neuroscience, and that is the brain is the body's most complex organ. And we turn our attention in this lesson to one of the most complex regions of that organ, the brain stem. So my learning objectives for you in this tutorial are for you to be able to identify the major subdivisions of the brain stem and the spinal cord, although our focus will be on the brain stem primarily. And I want you to be able to do so in representative transverse cross sections. That is, not just from looking at the outside, but from looking at the inside. And I want you to be able to have a discussion, maybe with a family member or a friend or a classmate. And I want you to discuss the relationship between the cranial nerves, that I trust you have some familiarity with now, and the nuclei that we find within the brainstem. And so what I'm referring to are the clusters of cells that receive sensory information from the cranial nerves, or the clusters of motor neurons that grow out the axons that run through cranial nerves on their way to muscle. Okay, so let me just remind you of the basic organization of our sensory and motor systems, beginning with our sensory system. So obviously we have information coming from the outside world and information coming from within our own body that is fed into the central nervous system via our special sensory cells that we find in our special sensory organs like the eye and the ear and the nose and the tongue. But we also have this vast sensory surface, our skin, as well as sensory cells that are growing out receptor endings in our muscles, in our joints, in our tendons, and all of this information informs the brain about the internal state of the body and the movements of the body through this environment. Well, all of this information is conveyed via axons that run in nerves, and those nerves are spinal nerves and cranial nerves. So these nerves are grown from cells, and those cells typically reside in ganglia, which are small clusters of cells just outside of the central nervous system. Now the special sensory organs are a bit different, and we'll talk about them in due course. So when we focus on the cranial nerves now, I want you to just have in mind a nerve which is very much like one of our spinal nerves, except these nerves are attached to the brain stem. All right, so these are sensory components that are fed up into higher levels of processing through pathways that we'll come to study one at a time over the next couple of weeks. So that's the sensory side. There's also a motor component to most of our nerves and most of our cranial nerves, and that motor component is concerned with activating skeletal muscles and activating muscles associated with the viscera, that is, smooth muscles, the muscles of the heart, cardiac muscle, as well as the secretory cells in our glands. These effector elements are engaged by axons that grow out of the central nervous system. And for skeletal muscle, for example, those axons are derived from motor neurons that reside in the ventral horn of the spinal cord and in a collection of nuclei in the brain stem. Now, the activation of smooth muscle, cardiac muscle, glandular tissue is just a bit more complicated. That gets us into a division of the motor system that we call the visceral motor system, or sometimes it's called the autonomic nervous system. And here we have a chain of neurons that connect the central nervous system to the effector cells that do the contraction, be it in the heart or in the blood vessels or in the viscera. So we'll talk about that topic in a few weeks from now. And hopefully the organization of that autonomic nervous system will be clear. For now, I just want you to get the big picture that the motor output is derived from two principal kinds of motor systems, a visceral motor system and a somatic motor system. And as we'll see, the somatic motor system can be further divided in terms of the embryological origins of the tissue that is innervated. Now, what we're leaving out from this diagram, of course, is the wonderful integration of sensation and the production of commands for motor output that takes place at higher levels of processing, namely in the cerebral cortex and in circuits that run through subcortical structures like the basal ganglia, the thalamus, and even the cerebellum. Well, all of this sensory motor integration will be the topic of forthcoming tutorials, so hang tight on that. All right, so with that as a background, let's now focus in on the cranial nerves themselves and the brainstem. So I will remind you of what the surface features of the brainstem look like, first beginning with the basic subdivisions of the brainstem. The brainstem is conventionally divided into a midbrain, a pons, and the medulla oblongata. all of which sits between the forebrain, which would include the thalamus and parts of the basal ganglia, which is basically what we have from about this region on in the anterior direction and the superior direction. And then below the medulla, we have the spinal cord. So the brainstem is an intermediate part of the central nervous system between forebrain and spinal cord. It's a very complicated place, and it includes most of what we call the hindbrain from embryological development of the nervous system. So the forebrain sits anterior and superior. The hindbrain is the brainstem, and the spinal cord is inferior or caudal to the brainstem. Okay, now let's look at the ventral surface of the brainstem and just quickly review the cranial nerves. And they're laid out for you in this figure from the textbook that we refer to. I would also refer you to my tutorial where I actually showed you the surface of the human brainstem in the lab. And I also have with me today the same model of the brainstem that we used. And I think rather than going through this figure, I'll just show you quickly this model. And if you feel like you're good on the brainstem and the superficial aspects of it, feel free to skip over this part. But otherwise, let's just take a few minutes and review the surface features of the brainstem. Well, I trust that you all can see this, and this brainstem is also produced for you and fully annotated and movable within Sylvius software. So if you have access to Sylvius, you might want to get that out and follow along and click along with me. All right, so what we're looking at is the ventral surface of the brainstem. This resembles the drawing that we had in the previous slide in the upper right. So from about this level on up is actually forebrain. So what we're looking at here, these large egg-shaped structures, that's actually part of the basal ganglia. Okay, and the hypothalamus and the thalamus on the dorsal aspect, which I'll show you more in just a moment. So the brainstem would be this region right in here. So we have the midbrain, the pons, the medulla oblongata, and just below we have the spinal cord. And I want you to notice the cranial nerves that are visible here in this model. Well, we've been talking mainly about the brainstem, so we're concerned with cranial nerves 3 through 12. Cranial nerve 1, the olfactory nerve, is not present in this model, and it doesn't attach to the brainstem. Neither does cranial nerve 2, the optic nerve. However, for completeness, we do have the optic nerve here in this brainstem model. And the nice thing about this model is that we can follow the optic nerve to the chiasm, and then we can turn the model around and note its progression from the optic tract right to the posterior pole of the thalamus, a region called the lateral geniculate nucleus that we'll talk more about in a later session. Well, that's cranial nerve 2. As I said, it does not attach to the brainstem, so we won't be talking about it further in this tutorial. But let's find the rest of the cranial nerves in this model. So cranial nerve 3 is right here. That's the ocular motor nerve. It's the nerve that emerges from the space right between these massive stalks that define the ventral surface of the midbrain. These stalks are the cerebral peduncles. The space between them is the interpeduncular fossa. And cranial nerve 3, the ocular motor nerve, emerges right in that fossa. Okay, cranial nerve 4 is a bit unusual. As you may recall, it's the trochlear nerve. It's unusual because it emerges from the dorsal aspect of the brain stem. It's the only motor nerve to do so. All the other motor nerves exit the ventral and lateral regions of the central nervous system. But here's the trochlear nerve. You can see it's a fine little thread that wraps around the posterior and lateral aspect of the caudal midbrain and then joins up to make its way to the orbit with the other ocular motor nerves. So trochlear nerve, nerve 4, nerve 5 is this very large protruding nerve, very obvious nerve. It plunges right through the fibers of the pons, the transverse fibers of the pons, so it's a very easy nerve to recognize in a whole brain specimen or in our illustrations. So that's the trigeminal nerve, nerve 5. Now next we come to the junction of the pons and the medulla, and along that junction are 3 pairs of cranial nerves. From medial to lateral, they are the abducens nerve, nerve 6, the facial nerve, which is the 7th cranial nerve, and then the vestibulocochlear nerve, the 8th cranial nerve. So 6, 7, and 8. If you notice where cranial nerve 8 is, just below it are the nerve rootlets of 9 and 10. So we have the nerve rootlets of the glossopharyngeal nerve, nerve 9, the rootlets of the vagus nerve just below the glossopharyngeal nerve. So the vagus nerve is cranial nerve 10. Cranial nerve 11 is a series of rootlets, which in the model, it looks like these wings that come off on either side of the lateral medulla, but really there's a series of rootlets that join up to form the spinal accessory nerve, cranial nerve 11. This nerve then enters the foramen magnum and then loops back out through the jugular foramen. So its source is actually in the upper part of the cervical spinal cord, but we consider it a cranial nerve nevertheless because it does enter the cranium. And then finally, nerve 12, the hypoglossal nerve, are these fine little rootlets here that emerge between the medullary pyramid along the midline and the olive, a bulging structure in the ventrolateral aspect of the medulla. So there's the hypoglossal nerve, nerve 12. Okay then, let's get back to our slide and make sure that you are comfortable and familiar with what we see here in this slide and that you know what the 12 cranial nerves are and the 10 that connect to the brainstem in particular for this lesson. Now notice that in this figure there's a color code, and the color code is meant to help you understand which nerves are purely sensory, which are purely motor, and then which nerves, like most of our spinal nerves, have a mixture of sensory and motor axons within them. Well, that may seem perhaps surprising that there would be nerves that are mixed for sensation and motor output, but that is the case for our spinal nerves, and it is the case, as you can see, by the abundance of green nerves here for several of the cranial nerves. So that suggests that there must be some complex relationship between the brainstem and the nerves, and often that is the case. So what I want to help you with next is to understand how do these nerves connect up with the gray matter within the brain stem that either are receiving incoming sensory signals or giving rise to axons that convey motor signals out to the effector tissues. Now, in order to show you what those gray matter structures are, we're going to be using a dorsal view of the brainstem, and that's what we have here. So we have removed the cerebellum. So the cerebellum normally would sit out here somewhere and receive input via these massive fiber bundles called the cerebellar peduncles. So we've taken those away, and what we're looking at now is the fourth ventricle, this space down here between the brainstem and the cerebellum. So we're looking into the floor of the fourth ventricle now. So I hope you can appreciate the midbrain, which is up in this region here, and then the pons between the midbrain and the medulla oblongata down below. Okay? So next what we're going to see is a phantom view of the brainstem with the cranial nerve nuclei that I want you to become familiar with illustrated. So this is something of a phantom view of the brainstem. We're looking through that dorsal surface through the floor of the fourth ventricle as if we could see the gray matter themselves. And what we see is a complex array of nuclei that come in different sizes and different shapes. And for the purpose of making this illustration just slightly less complicated than it needs to be, we've organized it by color code and by side. So on the left side of the figure, we have our sensory nuclei. And on the right side of the figure, we've illustrated the motor nuclei. Now, of course, the nervous system displays bilateral symmetry, so on each side we would have both the sensory and the motor nuclei. So imagine just folding this illustration of nuclei one side on top of the other so that all of these nuclei would exist on each side of the brainstem. And that's what we have when we look into the brainstem itself. Now, this illustration bears some resemblance to the dorsal side of the model that I showed you a moment ago. So let me show you that once again. So now we're looking at the dorsal side of the brainstem. Like the illustration, we've removed the cerebellum. We're looking into the floor of the fourth ventricle, and rather than making this a glass model, it's a rubber model. So what we've done here is dissected away the tissue to reveal the shapes and the bulges that indicate the location of cranial nerve nuclei. So I would encourage you to go into Sylvius, and you have a view of this dorsal aspect of the model with the nuclei identified. And there's also a feature in Sylvius that allows you to rotate this brainstem around so you can see these nuclei in various views. So I just wanted to show you what that looks like in the model, and now we can get back to the figure and recognize some of these important cranial nerve nuclei. Now, I do want you to make a pass through them all, but for this view I'll just highlight a few that might catch your eye in particular. One of them is this long structure in blue that has this sort of bulbous region right in the middle. This is the trigeminal complex of the brainstem. It can be divided into several subnuclei. We'll spend a fair amount of time talking about this particular complex of nuclei. It's all associated with the trigeminal nerve, and it's all associated with sensations derived from the face. But different parts of this nucleus receive different aspects of facial sensation. For example, this long pointy region in the superior direction, that's the mesencephalic trigeminal nucleus. There we find cells that are sensitive to the stretch in our jaw muscles in our temporomandibular joint. So you can imagine that must be very important in helping to govern chewing behavior. Well, in the middle, we have something called the principal, sometimes called the chief sensory nucleus of the trigeminal nerve. And this is the part that receives signals about light touch from the face. So if you ever have a gentle caress on your face, this is due to activation of cells in the trigeminal nerve that synapse in this principal trigeminal nucleus. And then lastly, and perhaps least pleasantly, we have this long descending part of the trigeminal nuclear complex. This is called the spinal trigeminal nucleus, and that's the part of the nucleus that's concerned with pain and temperature. So if you ever burn your tongue, burn your mouth on a hot cup of tea or a hot bowl of soup, or if you've ever had dental pain or some other source of pain in your face, you've activated this spinal trigeminal nucleus of your brainstem. And I'd like to just point out one nucleus on the motor side, just to whet your appetite for our studies of the motor system that we'll have in a few weeks. That's the facial motor nucleus, and we find it right here. So the facial motor nucleus is a collection of motor neurons that innervate the muscles of the face that convey expression. So whenever you smile, whenever you greet someone, whenever you laugh or cry or frown, or express some other human emotion, you're activating the outflow of your facial motor nuclei that are painting on your face the emotion within. Well, if you're feeling pretty overwhelmed right now, then join myself and the hundreds of thousands of students around the world in the last 100 years that have faced the challenge of understanding the complexity of the cranial nerve nuclei. So how can we possibly simplify this challenge for you? Well, I'm going to suggest one way to simplify it, and that is to use an embryological framework to help you understand this assortment of cranial nerve nuclei. Now in this figure, we notice that there are subdivisions of our color code where the various shades of red are indicating our different kinds of motor nuclei, and the shades of blue are indicating the sensory nuclei. And what these terms refer to are the embryological origins of the tissues that are connected up with these nuclei in the brainstem. So there are muscles that are derived from the embryological somites. So we call the axons that connect to those muscles somatic motor axons, and the nuclei that grew those axons somatic motor nuclei. And in the head and the neck region of the embryo, we have something called the pharyngeal arches. And from the pharyngeal arches are derived a variety of tissues, including some of the muscles of the cranial region and deep within the neck. And the motor neurons that innervate these derivatives of the pharyngeal arches that become muscles are called branchial motor neurons, giving rise to branchial motor axons. And so we can recognize a distinct set of nuclei in the brainstem that are connected to these muscles derived from the pharyngeal arches rather than the somites. So it's useful to distinguish the somatic motor from the branchial motor. And we'll do that as we look inside the brainstem. And finally, there are smooth muscle cells, of course, cardiac muscle tissue and glandular tissue that is innervated by axons that are controlled by the central nervous system. And that outflow from the CNS comes from our visceral motor axons. And those visceral motor neurons that grow those axons are found in the brainstem and also found in the spinal cord. And we'll get to the spinal cord in a later session, but for now I want to show you the brainstem nuclei that grew out those visceral motor axons. Now on the sensory side, we can also recognize different distinctions. There is a general sensory system derived from our skin surfaces, our muscles, our joints, structures in the peripheral parts of the body, so to speak. And then there are special sensory systems, our special sense organs, such as our eye, our ear, our tongue, our nose. So those are systems that are especially dedicated to processing just a fine modality of information derived from our environment. And then finally, there are visceral sensory axons that feed the central nervous system with information derived from our visceral organs. Well, as this color code makes explicit, these different submodalities for motor control and for sensation connect with different nuclei within the brainstem. And in the tutorial notes, and here in front of you now, is a really beautiful drawing made by my friend and colleague, Dr. Nell Kant, here in the Department of Neurobiology at Duke University. And it's a simplified way of explaining how one can relate the development of the neural tube to the more complicated, mature forms that we see in the spinal cord and in the brainstem. Well, we'll talk some about brain development in a few weeks, but for now, I would just emphasize that the nervous system is derived from the walls of a tube. And that's what we have here drawn simplistically in the upper left. So imagine this as a cross-section through the caudal part of the developing embryo where the nervous system is going to form the spinal cord. And I think, as you may know already, the dorsal part of that spinal cord is related to sensation. Sensory signals derived from the peripheral parts of the body or the superficial parts of the body, and then sensory signals derived from the viscera, the more interior organs within the body. That's all found in the dorsal aspect of the developing neural tube that will form the spinal cord. The ventral part, on the other hand, is motor. And again, there's a systematic organization where the motor neurons that innervate the tissues derived from somites, that is our striated skeletal muscles, they are innervated by cells that sit in the more the more ventral part of this developing neural tube, whereas the motor neurons that are going to supply motor signals to those ganglia that innervate the visceral organs, they come from this more intermediate region right in here. Now, that's the plan for the developing neural tube in that region that forms the spinal cord, and then when we actually look at what we see in the adult central nervous system, we see a spinal cord with this characteristic butterfly shape of gray matter in the middle, and we have a dorsal horn where we find our sensory neurons, and we find a ventral horn which is filled up with neurons that are affiliated with motor output. with this large anterior part of the ventral horn where we find our somatic motor neurons. Well, here's another way to conceptualize this. And I really want you to do all of this with me. You might even want to stand up if you're comfortable doing that in a position to do this. So I want you to take your two hands and I want you to put them together in such a way as they resemble the illustration that we have here in the upper left. Okay? Let me clear our annotations so that you can see that clearly. So what I want you to do is I want you to make with your hands this figure. Okay, so here we go. We have just made a cross section of the developing neural tube. Now, I want us to note how our hands resemble the distribution of these neurons related to sensation and motor control. We have our fingers up on the dorsal aspect of this transverse section. The fingers would represent our somatic sensory neurons, whereas the palms of our hands are sitting in a more anterior or ventral position. The palms of our hands would represent our somatic motor neurons. But we also have these knuckles, right? Near the junction of our palms and our digits. So the knuckles would represent the visceral elements, with the visceral sensory elements right on the top of our knuckles and the visceral motor elements just on the anterior or ventral aspect of the knuckles relative to this cross-section. Okay, so we've got somatic sensation, dorsal, somatic motor control, ventral, and then along the ridges formed by our knuckles we have visceral sensory and visceral motor. All right, now let's take this conceptualization up into the brainstem. So what happens in the brainstem is that the ventricular system expands. In the spinal cord we have a central canal, a central space for the flow of cerebrospinal fluid. But in the brainstem we have a large fourth ventricle. So essentially this space expands in the following way. The sensory elements open up to form the floor of the fourth ventricle, with the digits now still representing somatic sensation, but now they are lateral underneath the floor of the fourth ventricle. And where would we find our somatic motor cells? Well, we'd find them in the position of my palms, near the midline, in the dorsal part of the tegmentum, just underneath the fourth ventricle. Now notice where my knuckles are. My knuckles are somewhere between the somatic motor region and the somatic sensory region. And that's where we find our visceral sensory and our visceral motor neurons. So again, a very simple neural tube opens up to form a fourth ventricle, with the sensory systems moving far lateral. All right, now that simple picture, hopefully, will stay with you. Now, if you did that with me, hopefully the significance will stay with you, and you'll have a picture of the developing brainstem, where our somatic sensory elements, and we'll also add now our special sensory elements as we get into the cranial region, are to be found in a far lateral position. And just medial to those, we would find our visceral sensory and special sensory systems related to the viscera, specifically our sense of taste. So these sensory elements are lateral. Now more medial, we find the motor elements right along the midline of the tegmentum underneath the ventricle. We would find our somatic motor cells, and just between the somatic motor and the sensory cells we'll find our visceromotor cells. Now when it comes to the cranial region, there are different kinds of muscles, right? So in addition to the smooth muscles and the striated muscles, there are striated muscles that are derived from somites and those that are derived from the branchiomeric arches, or the pharyngeal arches of the developing embryo. These include many of the structures around the cranial region and into the neck region, and there are different motor neurons that are growing out axons to innervate either the striated somatic somite-derived muscle or the striated muscles derived from the branchiomeres. And those that are derived from the somites are receiving input from our somatic motor cells. which are found again right along this dorsal midline underneath the floor of the fourth ventricle. The motor neurons that supply innervation to the derivatives of the branchiomeres have migrated into an intermediate position. So these motor neurons initially sat very near our somatic motor neurons, but they migrate into a more intermediate, somewhat more anterior and lateral position. And as they migrate, their growing axon is dragged down with it. So consequently, we have motor neurons, such as those that supply the muscles of facial expression, that sit in this intermediate position and grow axons that loop around in this fashion. And in this case, what I'm describing for you is the facial motor nucleus and the facial nerve. Okay, now I think we're ready to apply this embryological framework to understand the organization of the cranial nerve nuclei in the brainstem. So this is a chart you might want to have in front of you for a little while so that you can begin to make sense of all of this new anatomical information that I'm sharing with you in this tutorial. So what this chart does is it organizes the brainstem according to embryological division, that is midbrain, pons, and medulla, and also organizes across our various ways of breaking down motor outflow and sensation according to our embryological framework. So we have 6 columns in this chart. We have 3 to the left of center for motor control and 3 to the right for sensation. And for motor control, we recognize a somatic motor division, a branchial motor division, and a visceral motor division. So here's how I want you to think about this. I want you to think about each embryological division of interest for motor control and for sensation, and consider what's in the midbrain, what's in the pons, what's in the medulla. So for example, let's think about the motor control for the muscles that are in the cranial region that are derived from somites. And it's fairly simple. It's the muscles that move the eyes in the orbit and the muscles that move the tongue. So the muscles that move the eyes are related to cranial nerves 3, 4, and 6. Cranial nerves 3 and 4 are found in the midbrain. Cranial nerve 6 attaches to the junction of the pons and the medulla, but the nucleus that grew out those axons is found in the caudal pons. The axons that move the tongue are derived from the hypoglossal nucleus, which is found in the rostral part of the medulla. So we have the hypoglossal nucleus. So one way to approach this challenge is to consider how the outside of the brainstem is related to what we find on the inside. So again, taking our somatic motor nuclei as an example, we have the nerves that move the eyes, the ocular motor nerve, the trochlear nerve, and the abducens nerve. 2 nerves associated with the midbrain, 1 associated with the caudal pons. Up here in the midbrain, we have our ocular motor nucleus growing out these axons to move the eyes, the trochlear nucleus growing out the axons in the 4th nerve, and the abducens nucleus growing out the axons in the abducens nerve. Now, the muscles derived from the pharyngeal arches, we call those branchiomeres or branchiomeric muscles, and the motor nuclei that innervate those muscles are called the branchial motor nuclei. And we just think about, what are they? Well, they are the muscles of mastication, that is, the muscles involved in chewing. They are innervated by a motor component of the trigeminal nerve. There's the muscles of facial expression that are innervated by the facial nerve. So these axons are grown out by the facial motor nucleus. So both the trigeminal motor nucleus and the facial motor nucleus are going to be found in the pons. Now, a little less obvious, but important nevertheless, are the muscles that are deep within our neck region, around our larynx and our pharynx. Those muscles are controlled by components of cranial nerves 9 and 10, the glossopharyngeal nerve and the vagus nerve. And the axons that are governing these muscles, deep in the larynx and the pharynx, are grown from neurons in what's called the nucleus ambiguus, for reasons that I think will be obvious to you shortly. And then finally we have our spinal accessory nerve. The spinal accessory nerve innervates also muscles derived from the pharyngeal arches that are involved in turning the head and shrugging the shoulders. That would be the sternocleidomastoid muscle and part of the trapezius muscle. Now let's move on and think about our visceral motor elements. So within our eyes, we have a smooth muscle in the iris that constricts the pupil. And that constrictor function is derived from parasympathetic neurons that send their axons out through the ocular motor nerve. And these parasympathetic preganglionic axons live in a small nucleus that sits right on top of the ocular motor complex. That nucleus is called the Ettinger-Westphal nucleus. So that is our most superior visceral motor nucleus in the brainstem. Deep in the pons, and we won't see them here in this view from the outside, are the nuclei that are responsible for the secretion of saliva and also the secretion of tears from the lacrimal gland. So we have these salivatory nuclei that sit deep in the pons, and they send axons out through cranial nerves 7 and 9 to their respective targets in the orbits and in the oral pharynx. Also in the junctional region near the pons and the medulla, we have the neurons that are associated with cranial nerve 10, the vagus nerve, that control the viscera that are found in the thorax and the upper part of the abdomen. So these axons are grown out by the nucleus ambiguus and by the dorsal motor nucleus of vagus. Now notice that we've named the nucleus ambiguus twice, and I don't want you to be confused about why that is the case. There is some historical reason why we call two different sets of neurons that convey two different modalities of motor control part of the same nucleus, but they are not the same cells that supply the pharynx, larynx, on the one hand for the branchial motor control, and the cardioinhibitory output that slows the heart via the vagus nerve on the visceromotor side. So two different populations of neurons that sit within the same nuclear complex of the upper part of the medulla. Moving on now to the sensory side, we have our general sensory signals that are coming from the skin surfaces of our face and our head, and some of the deeper structures associated with the muscles of our face. These are giving rise to signals about light touch, about movement, about pain and temperature sensitivity, and all of this information is conveyed to the central nervous system across 4 cranial nerves 5, 7, 9, and 10. And all of this information is fed into that long trigeminal nuclear complex. And if I look again at the brainstem model, we see a long nucleus that seems to just snake its way down from the midbrain or the mesencephalon through the pons and into the medulla. That's our trigeminal nuclear complex. And so the mesencephalic portion of it, that is the midbrain portion of this is where we find our movement-sensitive somatic sensory signals related to the movements of muscles in our face, especially the movements of our chewing muscles and the movements of the temporomandibular joint. The sensory signals that are derived from our skin surfaces pertaining to light touch and other aspects of mechanosensation are fed into what's called the principal nucleus of the trigeminal, which is that large bulbous structure in the pons. And these signals are derived mainly from the trigeminal nerve, but also a small contribution related to a little patch of skin in the external ear that's conveyed across cranial nerves 7, 9, and 10. And then lastly, we have the spinal trigeminal nucleus, and this is the inferior or caudal extension of this nucleus that receives pain and temperature information. We'll talk much more about both of these two preceding divisions of the trigeminal system as we get into a discussion of mechanosensation and pain and temperature sensitivity. Okay, well, this leaves us then with a brief consideration of our special sensory systems and then our visceral sensory systems. Our special sensory systems that are connected to the brainstem are those related to our sense of hearing, our auditory system, and our sense of balance, being derived from the vestibular system deep within the petrous temporal bone. So both of these two special sensory systems are sending signals to the brainstem via the 8th cranial nerve, and those signals diverge once they enter the brainstem into different sets of nuclei. There's a set of nuclei related to hearing called the cochlear nuclei, and then a set of nuclei related to our sense of balance, equilibrium, basically the accelerations of our head and the static positions of our head. That's the vestibular nuclear complex of the brainstem. Both of these nuclei are found near the junction of the pons and the medulla. And finally, we have our visceral sensory signals that enter the brainstem via cranial nerves 7, 9, and 10. We can divide them into special visceral sensory, meaning basically our sense of taste, and that sense innervates the rostral part of this nucleus of the solitary tract. The other aspect of visceral sensation is more of a general sense of visceral sensation that is conveyed to the same nucleus but to a caudal division of it. So the nucleus of the solitary tract is all about visceral sensation. I'll just put this table in front of you again, which runs through the cranial nerves and gives you a sense of where the nuclei are to be found and what they are as they relate to these cranial nerves, and also says a word about how one might think about testing the function of these nerves. So you might want to take a moment at some other point, come back to this table, and maybe try to do some of these tests on a friend or a family member. They're pretty easy to do. They don't really require any special equipment, and if you just think about the actions of our muscle systems and the activities of our sensory systems, I think you can very readily figure out how a clinician might go about testing the integrity and the function of each of these cranial nerves. And if you want to see how a clinician might do this, I would refer you to an excellent website that is maintained by a fantastic neurologist and author, Dr. Hal Blumenfeld. And that website is neuroexam.com. So check it out. You'll see lots of cranial nerves being tested in the standard way that a neurologist would do it, so I would encourage you to have a look at that. neuroexam.com. Okay, finally, we are ready to actually look into the brainstem and find these cranial nerve nuclei. So we can do that by looking at some cross-sections, and the figure in front of you here shows cross-sections through various levels of the brainstem, beginning at the top of the brainstem with the midbrain. So in a section through the midbrain, we should be thinking about, okay, what are the nerves that connect to the midbrain again? Remember, nerve 3, nerve 4. So nerve 3 is in a more superior position, and that's what we find in this first slice. We find the oculomotor nucleus and the Edinger-Westphal nucleus, both growing axons out to the third nerve. Now, not illustrated here because we'd have to be right at this level would be a view of the trochlear motor nucleus, but we'll see that in a few minutes. Now, below into the pons, at two different levels of the pons, we first find the principal trigeminal nucleus and the trigeminal motor nucleus sitting right next to one another at the level at which the trigeminal nerve actually sends its axons into the brainstem. So that's the level at which we have this slice number 2. Slice number 3 is just below it, and the purpose of catching this slice is to show you the facial motor nucleus, which as a branchiomeric motor nucleus is just a little bit off of the dorsal aspect of the tegmentum because of the anterior or ventral migration of these motor neurons in development. So that's where we find our facial motor nucleus. We also have some nuclei associated with sensation here, the spinal division of the trigeminal nucleus. In red here we have the somatic motor nucleus, the abducens nucleus, and then lastly the vestibular nuclei that we're beginning to see as we get into the caudal part of the pons. Now we go just a little bit further down near the junction of the pons and the medulla. We find additional vestibular nuclei in about the same position, but we also see the cochlear nuclei, which seem to sort of cap the dorsolateral aspect of the upper medulla. We're continuing to progress through the caudal medulla, so we would expect to see the spinal nucleus of the trigeminal complex, which we have here in this blue shape. It's extending all the way down through the medulla, and in fact will seem to merge with the upper part of the spinal cord as the spinal nucleus of the trigeminal complex merges with the dorsal horn of the spinal cord. Well, there are a number of other nuclei to note here in the tegmentum of the medulla, all in that dorsal region. And again, as we look at this, I want you to recognize that the embryological framework helps us to interpret this. So right along the dorsal midline, we're going to expect to find a somatic motor nucleus. That's where we find the hypoglossal nucleus. Now, just to the lateral side of that somatic motor nucleus, we might expect to find a visceral motor nucleus, and we do. We find the dorsal motor nucleus of vagus. All right, now just lateral to our motor nuclei, we should expect to get into sensory nuclei, and indeed we do. First, we find the nucleus of the solitary tract, which is found right there in purple shade, and just lateral to it we find our special sensory nuclei, the vestibular nuclei. So again, motor nuclei are medial, sensory nuclei are lateral. And then finally, in the far lateral region, that's where we find our trigeminal system. Okay, and finally down into the section number 6 that we see here in this illustration, we have the caudal part of the medulla getting close to merge with the upper part of the spinal cord, and we essentially see the same set of cranial nerve nuclei, just in slightly different positions and somewhat reduced in size, with the exception of the spinal trigeminal nucleus that continues to be a prominent feature of the medulla throughout its entire length. Okay, so what I'd like to do with you next is to get into Sylvius and show you what these nuclei actually look like in histological sections taken through the brainstem. So if you're ready for a break, this would be a good time to pause and open up Sylvius and get ready to perhaps go back and forth between the remaining segment of this tutorial and Sylvius, should you have it. If you don't have Sylvius, that's fine. Just refer back to my tutorial notes as well as these images in this movie file, and follow along as I give you a tour of the brainstem through our histological atlas that we have in Sylvius. Okay, welcome back to this tutorial on the cranial nerve nuclei. If you have Sylvius 4 software, go ahead and open it and follow along with me. If you don't, that's fine. Just enjoy the tour. Maybe if you have Sylvius, you may just want to watch the remaining segment of this tutorial and then go back and repeat what I do and do it on your own time in your own way. So however you like, let's begin by getting into Sylvius 4, and I'm going to open up the Brainstem Cross-Sectional Atlas, and I'll just click on All Structures. So this opens up the atlas within Sylvius 4 that's devoted to the brainstem and the spinal cord. And the default here is to show us the very top section, which, as the navigator tool indicates, is taken from the upper part of the brainstem. So this is really the junctional region between the thalamus and the brainstem. We recognize the brainstem by these prominent cerebral peduncles that are taking shape, but we know we're still in the caudal part of the diencephalon because we have the mammillary bodies present along the ventral midline. So let's just get down one section below, And now this is looking more like the midbrain because we see these massive cerebral peduncles on the ventrolateral surface. We see a small cerebral aqueduct that tells us that we're cutting through the region of the brain called the midbrain. Now, what's attached to the midbrain? Well, the oculomotor nerve. So here's the oculomotor nerve, and we can follow its roots back towards a somatic motor nucleus along the dorsal midline and a visceral motor nucleus that we should expect to find sitting very near it. And indeed, if we follow these axons back, we have the oculomotor nuclei, which are the somatic motor nucleus of the third nerve, and then just superior to the oculomotor nucleus is our Edinger-Westphal nucleus, the source of the parasympathetic outflow through the third cranial nerve. Okay, going just a little bit more inferior now, we are near a region where the midbrain is beginning to join the pons. So we still see some cerebral peduncle, but now we're looking at some nuclei and some fibers that are associated with the pons. So we know we're right near that junctional region. That's very near the region where the trochlear nerve emerges from the posterior part of the brainstem. And indeed, here is our trochlear motor nucleus. Again, what would we expect to find? We would expect to find a somatic motor nucleus near the dorsal midline of the tegmentum of the brainstem. So there's our trochlear motor nucleus. Very small nucleus, and the trochlear nerve is a very small nerve. So that's nerve 4. Nerve 5 is near the center of the pons, so we need to section into the pons a little bit. So we need to section through the pons just a little bit in order to get to the trigeminal nerve. And at this level, we see these broad, massive nerve roots of the trigeminal nerve on either side. And if we follow these nerve roots back, they connect to a set of nuclei. So more lateral is where we find our chief sensory nucleus of the trigeminal complex, also called the principal sensory nucleus. And just to the medial side of that nucleus, we would find the branchial motor nucleus, the trigeminal motor nucleus. Now, the mesencephalic nucleus actually begins at about this level. It's found right about in this region. Very small nucleus, but if I select it, we can recognize this nucleus as it runs in the superior direction, just along the lateral aspect of the gray matter that is just on the lateral margin of the fourth ventricle as it begins to close off into the cerebral peduncle. So this nucleus really just trails off as a thin collection of cells that sit just medial to the tract of this nucleus, which are the axons feeding this nucleus. So right in this position is where we find these cells that give rise to the mesencephalic division of the trigeminal. Okay, well, let's get back to the trigeminal nerve. So here again is the trigeminal nerve, the chief sensory or the principal nucleus of the trigeminal complex, and the trigeminal motor nucleus. Now at this level, we are getting very close to those cranial nerves that we would find along the border between the pons and the medulla. We're not quite there yet, but we're getting close to it. So if we go down one section, now we're beginning to see some of those nerves and nuclei. So what are those nerves again? It's nerves 6, 7, and 8, the abducens, the facial, and the vestibular cochlear. So the abducens nerve is connected to the abducens nucleus, a somatic motor nucleus here along the dorsal midline of the tegmentum of the pons. And we can actually see some of the axons of the abducens nucleus that are growing out in the ventral direction to exit right about here at the junction of the pons and the medulla, pretty close to the midline. Now, also in this level of the pons, we have our facial motor nucleus, and this is another branchial motor nucleus, so it's not going to be along the dorsal margin of the tegmentum, but set back in the intermediate and lateral region of the tegmentum due to the embryological migration of these branchial Merrick motor neurons. So here's our facial motor nucleus, and the facial motor nucleus gives rise to axons that do something very strange. They actually sweep across the top of the abducens nucleus. So there is our nerve root of the facial nerve and the facial motor nucleus itself. So the axons didn't exactly grow up and around. Actually, the neurons themselves migrated in the anterior and lateral position, and they simply elongated the axon as they migrated down. So these cells used to be up here somewhere, but they ended up migrating down, and with it they drew out their axons. So the seventh nerve ends up exiting the brainstem in this lateral position out here. And just to the lateral side of the facial nerve, we expect to find the vestibulocochlear nerve, which is right in this domain here. So the vestibulocochlear nerve is related to a set of nuclei called vestibular nuclei. We see several of them here in this cluster near the lateral aspect of the tegmentum of the pons. We don't yet have the cochlear nucleus. That's found in a slightly more inferior position in the upper part of the medulla, so we'll get to that in just a moment. Now also in the lateral aspect of this tegmentum, we would expect the sensory nuclei associated with the trigeminal complex. And now that we are below the trigeminal nerve, we're going to be below that chief sensory nucleus. What we find is our spinal nucleus of the trigeminal complex. And just to the lateral side of that nucleus are the axons from the trigeminal nerve that supply the nucleus. We call this the spinal trigeminal tract. And then just to the medial side of the tract is the spinal trigeminal nucleus. Okay, now let's go down one section. And now we're in the upper part of the medulla. The section looks a lot bigger, but really it's a magnification of the section for clarity purposes, not to scale. So the brainstem's actually getting a bit smaller now that we go from pons to medulla, but our magnification is actually increased. Now, unfortunately, we didn't have an optimal section to put into Sylvius to demonstrate the cochlear nuclei, so they're not labeled, but they're present here nevertheless. So notice that there's this big dark bundle of white matter here called the inferior cerebellar peduncle, and I say dark because this is an image of sections through the brainstem where the white matter is made to look dark and the gray matter is made to look light. I should have said that right from the start. I'm sorry about that. But just to clarify, the white matter is dark, the gray matter is light. So this is a big bundle of white matter in the dorsolateral aspect of the medulla that we'll talk about in a later session. This provides signals from the spinal cord to reach the cerebellum. Just to the lateral margin of that bundle here in the upper medulla, we see some gray matter, which is unstained in this view of the brain. And so this gray matter, that's part of the cochlear nuclear complex. In fact, this little tag of tissue here, that's a bit of the eighth nerve. So the cochlear nuclei are in the lateral margin just outside that inferior cerebellar peduncle. The vestibular division is just on the medial edge of this inferior cerebellar peduncle. So here we have a couple of divisions of the vestibular nuclear complex. They would connect up with axons that are coming right here from this vestibular cochlear nerve. Now, we are also beginning to encounter the rostral part of this very interesting visceral sensory nucleus called the nucleus of the solitary tract. So here's the nucleus. There is something called the solitary tract. It's actually the axons from nerves 7, 9, and 10 that supply signals into this nucleus. So there's the nucleus, and if we go down one section, we see this a bit more distinctively. There's something like a bullseye shape where the nucleus is surrounding the tract. Kind of an unusual configuration if you can compare that to the situation we have with the trigeminal system. In the trigeminal system, we have the tract that sits out here lateral to the nucleus, but in the solitary system, the tract is right in the middle of the nucleus, so the gray matter actually surrounds the nucleus with respect to the solitary tract. So it's an interesting shape. It's a visual distinctive as we look into a section through the upper part of the medulla that is stained to resemble a myelin stain where white matter is dark and gray matter is light. Okay, so also here in the upper medulla, you should be thinking about a somatic motor nucleus, the hypoglossal nucleus. We would expect to find it along the dorsal margin. of the tegmentum, and there's our hypoglossal nucleus. Now, if you'll remember where the hypoglossal nerve roots exit the brainstem, they do so right along this cleft between the medullary pyramid and the olive. So we can imagine if we took a section through here, what would we see? We might expect to see fibers emerging between the pyramid and the olive. And that's exactly what we see here in this section. So there's the hypoglossal nerve, and we can follow its trajectory right between the medullary pyramid, which now we see in cross-section, a dense bundle of white matter, and the olive. And the olive is made from this fantastically looking gray matter nucleus called the inferior olivary nucleus, and some white matter that's on the outside of that nucleus. So we'll talk about the inferior olivary nucleus when we talk about cerebellar function. Really a fascinating story about how this inferior olivary nucleus induces plasticity in the cerebellum. But for now, we'll just use it as a visible landmark that allows us to know where to expect to find the emergence of the 12th cranial nerve between the olive and the pyramid. All right, let's get back up to the dorsal tegmentum. We talked about the somatic motor nucleus on the midline. We talked about the visceral sensory nucleus that is a bit more lateral, and our special sensory nuclei, the vestibular nuclei, and then our general sensory nucleus in the far lateral region. But between the motor and the sensory, we might expect to find some visceral motor. And indeed, we have a major visceral motor structure here called the dorsal motor nucleus of the vagus nerve. So this is where many of our parasympathetic visceral motor cells reside that grow out axons through the vagus nerve. Also contributing to that vagus nerve are parasympathetic neurons that are involved with inhibiting the rate of contraction of the heart and the contractility of the ventricle. Those parasympathetic visceral motor neurons come from this obscure, hard to see, ambiguous nucleus in the medulla called the nucleus ambiguus. Sometimes it's nice when the names tell us something about what we expect to see, when this is one such case. So here's the nucleus ambiguus. As I mentioned, it sort of has two parts to it. There's the cardioinhibitory parasympathetic outflow, but then there's also the brachiomotor outflow, which goes to the muscles of the pharynx and the larynx, also via cranial nerves 9 and 10. Okay, well, let's just follow down a few more sections, and we'll get below the level of the hypoglossal nucleus and nerve here. What we will encounter, however, is the spinal trigeminal nucleus, and we'll see that we still have a tract to the lateral side of the nucleus, and as we go through the brainstem, at least in Sylvius, we use anatomical convention to recognize different subdivisions of this spinal trigeminal nucleus. There are really multiple subdivisions that have different functions in the sensations of pain and temperature derived from the face and the oral cavity. We won't get into all of that now, but I'll just point out to you that that's why we see these various divisions in Sylvius here. Now, these divisions eventually seem to run right into the dorsal horn of the spinal cord. So there's this bright compact part called the substantia gelatinosa, and I want you to notice as we get up into the brainstem, there's also a bright compact part of the spinal trigeminal nucleus that we call the gelatinosa part. So again, very much parallel organizational system to what we see in the dorsal horn of the spinal cord, which, as we'll come to see in a forthcoming tutorial, also is concerned with pain and temperature sensation from the body. Well, I hope you enjoyed that brief tour of the brainstem and just a little bit about the spinal cord. We'll have more about the spinal cord in a later session. I would encourage you to go through the tutorial notes, find those cranial nerve nuclei that are highlighted in bold font, and then get into Sylvius and find them here. And if you have any trouble, I'll just remind you that we have a glossary feature built into Sylvius, which will give you a chance to look up a term that might be of interest to you. So for example, let's just type in trigeminal, see what happens. We find a number of targets come up if we want to see the trigeminal nerve. We have an opportunity to view it in different views, including the view of our brainstem model here. If we want to find a particular nucleus, such as the spinal trigeminal nucleus, again, we have different views, some of which are included in the tutorial. Others are unique to Sylvius. This one in particular you might enjoy. This is an animation that illustrates for you how pain and temperature signals get from the trigeminal nerve into the brainstem, and the sounds that you heard are an indication of the synaptic junctions between one axon and the next neuron in the pathway. I bet you didn't know synapses made sound. Of course, I'm joking. Synapses don't make sound, but we put them in this animation just to highlight the fact that there is a junction from one neuron to the next neuron. And it will be important for you to be able to speak about the pathways with the precision of language that conveys your understanding of the locations of cell bodies, of axons, and synaptic connections within the pathways. So we'll be getting to that in the next few tutorials, which will turn our attention from the gray matter structures to the pathways that allow these gray matter structures to be interconnected into a sensory system. So I look forward to talking to you now in more depth about our sensory systems, beginning with our capacity for mechanosensation. So I'll see you next time. Hello again. Welcome to this tutorial on the internal anatomy of the spinal cord. Once again, we're confronted with the complexity of the nervous system, so our core concept is that the brain is the body's most complex organ. And our learning objective for today is that I want you to be able to identify the major subdivisions of the spinal cord as seen in representative transverse cross-sections. So this requires enough familiarity to the internal anatomy of the spinal cord that we can view in cross-sections so that you can understand what level of the spinal cord is in view and what are the important gray matter and white matter structures that we can find there. All right, well, let's begin by looking at a histological section of the spinal cord. This happens to be from the lumbar enlargement of the human spinal cord. And this is a section that's been stained for the presence of Nissl substance. Nissl is a term we use in neurohistology. It refers to rough endoplasmic reticulum. So wherever we have rough ER within the cell, which is typically in the cytoplasm, and there's some rough ER within the nucleus as well, but mainly in the cytoplasm, and perhaps into the proximal dendrites. That's where we will find the stain. So this is going to be a stain for cell bodies and for neurons, for also the proximal portions of the dendrites. So what's not stained here are myelinated axons. So this is one way of looking at the nervous system that highlights the presence of cell bodies. And what we can see in this image is that within the spinal cord we have gray matter that is central to the cord, and that gray matter is surrounded by white matter. So white matter is found all in these peripheral regions on both sides of the spinal cord. Okay, now let me give you some terms that will help you as we go and look at the structure within the spinal cord. So the white matter that we find on the dorsal aspect, and this is dorsal, is called the dorsal column. The white matter that we find on the lateral sides of the cord is called the lateral column. And the white matter that we find in the ventral or the anterior parts of the cord is called the ventral column. So this word column appears repeatedly in our lexicon that we use to describe white matter in the spinal cord. So when you see the word column, think white matter. Now, occasionally we apply the word column to talk about the organization of cell bodies, but I'll try to be very clear when that's the case. Otherwise, when you see the word column in your readings or in our discussions, think about white matter. Okay, So now let's turn our attention to the gray matter of the spinal cord, and we can recognize basically two broad regions of gray matter in the spinal cord. There's something that we call the dorsal horn, which can be found in this aspect of the gray matter of the spinal cord, and then there is a ventral horn. And the ventral horn then would be pretty much the rest of this, excluding perhaps what we might consider to be an intermediate zone between the dorsal horn and the ventral horn. We'll come back and talk about that as we get into different levels of the spinal cord. But the dorsal horn and the ventral horn provide us with our basic dichotomy between a sensory zone of gray matter, which is what we find in the dorsal horn, and a motor zone, which is what we find in the ventral horn. So in the dorsal horn, we have longitudinal layers of cells that are organized along the long axis of the spinal cord, one layer on top of the other, almost like a long series of strips of cells. And these strips have been recognized by anatomists over the years, and the scheme that seems to has stuck the best was proposed by an anatomist by the name of Rexed. And we call this scheme Rexed's layers, or Rexed's laminae of the dorsal horn. So that's what the Roman numerals are over here to the left side. So the dorsal horn is essentially laminae 1 through 6. We'll spend some time talking about these laminae when we talk about mechanisms of pain and temperature processing, especially lamina number 2, also known as the substantia gelatinosa, or the gelatinous layer of the dorsal horn. So we'll come back to that in a later session. For now, I just want you to get the idea that the dorsal horn is gray matter that is receiving incoming sensory signals from nerves that are running in through the dorsal roots and synapsing upon cells in the dorsal horn. Now let's turn our attention to the ventral horn, which is where we find our lower motor neurons, that is the motor neurons that innervate muscle, and the local circuit neurons that coordinate the output of those lower motor neurons. And here we have less of a layered organization to these cells, more of a column organization to the cells. So for example, we may find a column of cells that all innervate the same muscle. Not necessarily the very same muscle fiber, but perhaps a distributed set of muscle fibers that are within the same anatomical unit that we would recognize as a muscle. So if the dorsal horn is organized into long strips of cells, then the ventral horn is organized more into columns. And so here in lamina 9, we actually have something more like a rod shape of cells. And there are other more rod shapes, collections of cells that we recognize as laminae 7 through 9. Now we still use the term laminae, but as I'm suggesting, the organization in the ventral horn is more columnar or rod-shaped than it is laminar. All right, so there are connections between the dorsal horn and the ventral horn made by local circuit interneurons, and in one special case, there's an incoming sensory axon that synapses directly on alpha motor neurons. That mediates our very fast myotatic reflexes. So we'll talk about that in a later tutorial. For now, I just want you to have an overview of the organization of the gray matter in the spinal cord. And since it's gray matter, we expect to see cell bodies, and that's why I'm showing you this Nissl stain. But now let's turn our attention to the white matter that surrounds that gray matter. And for that purpose, I want to show you a myelin stain section. So this is a typical way that a neurohistologist might look at a cross-section through the nervous system. One can often see great detail that is otherwise not made visible in a Nissl stain if you stain with silver salts or perhaps gold chloride, or otherwise make the white matter look dark. So please don't be thrown off by this convention. Essentially, wherever we see dark staining, we are looking at white matter. So that means that where we see the lighter stain, that is going to be gray matter. So this is essentially the inverse visual appearance of what we just looked at with the Nissl stain. Okay, I think that should be clear enough. So what we have here is white matter that surrounds gray matter. And we recognize basically 3 zones of white matter around this gray matter. On the dorsal side of the spinal cord, or the posterior side, we have the dorsal column of white matter. On the lateral side, roughly to about this region, we have what we call the lateral column. And then on the ventral, or the anterior side, we have white matter that we recognize as the ventral, or the anterior column of white matter. Now, in each region of white matter, we're going to find different kinds of sensory or motor pathways. And we'll get into that in quite a bit of detail as we progress over the next couple of weeks. So I won't give you all that detail now, except to just make a few broad statements about how these tracks are organized within this white matter. Essentially, the dorsal column is a sensory zone. There are sensory axons that are ascending the spinal cord in the dorsal columns. In the lateral column, we have a mixture of sensory axons and one very large and very important motor pathway that occupies actually most of this lateral column, and that's the lateral cortical spinal tract. We'll spend a fair amount of time on the organization of that one pathway. Now, likewise, on the ventral, or the anterior column, we have a mixture of both sensory and motor pathways. And here, with respect to the sensory pathway, there's one very important pathway that we'll spend a fair amount of time talking about. And that pathway we find roughly in this region of the ventral or anterior column. And this is where we find our pain pathways that are ascending the spinal cord, conveying sensations about pain and temperature to processing stations in the brainstem and in the forebrain. Much of the rest of this system is a collection of small descending motor pathways that are conveying signals mainly from the brainstem to the ventral horn of the spinal cord. All right, so stay tuned. We'll talk about those sensory and motor pathways in detail as needed as we get into a discussion of sensory motor systems. Now, with some overview of the white matter and gray matter of the spinal cord, I want us to turn to a very important consideration, and that is understanding what we find at each level of the spinal cord. And here's, I think, a very helpful figure. What it does is it shows representative cross-sections that are taken from different levels of the spinal cord. And I want you to notice a few features that we find here. Essentially, what we find is that in the region of the cervical spinal cord and also in the lumbosacral enlargement region, we see an expansion of the amount of gray matter that's present in the ventral horn. I think that should make good sense to you. This gray matter is providing motor output to the arms, in the case of the cervical cord, and to the legs, in the case of the lumbosacral enlargement. So more neurons, more local circuits means more gray matter volume that's governing what we do with our appendages, our arms and our legs. In the thoracic spinal cord, notice that the amount of gray matter in the ventral horn is reduced compared to what we find in the cervical and in the lumbar and sacral levels. Okay, so look at how much gray matter we have in the ventral horn. That will help you identify the section. I also want you to notice the amount of white matter that we have here in the spinal cord. Now, for the amount of white matter in the cord, I think it should make a certain amount of sense that it would decrease as we move from superior to inferior. And it's decreasing for 2 reasons. One reason is that the axons that are coming down from the brainstem and cerebral cortex in the lateral and anterior column of white matter are simply terminating along the way. So if we looked at the upper levels of the spinal cord, we would expect to find more axons than if we were to look at the lower levels of the spinal cord. Now, for a similar reason, our ascending pathways that are sending signals, let's say about pain and temperature or mechanosensation up through the spinal cord, are collecting at various levels, and as a consequence, in the superior segments of the spinal cord, we have more axons conveying ascending sensory signals than we have at more inferior levels. Now, if we then look at the cross-sections of the spinal cord, I think it should be obvious to you that we have a whole lot more white matter here in the cervical cord compared to the very bottom of the spinal cord in the sacral region. So look for the amount of white matter that we find, and we see that there is more white matter at the upper levels than in the lower levels. Now, one key difference that I want you to look for is the lateral margins of the gray matter of the spinal cord. You'll note that in the thoracic cord, there is a lateral protrusion from the gray matter right near the junction of the ventral horn and the dorsal horn. This is a feature that we call the lateral horn. And this is a distinctive feature of the thoracic spinal cord. And as we'll see, what's in that lateral horn are preganglionic sympathetic neurons that are providing outflow from the nervous system to the viscera. We see a similar group of cells in the sacral cord, but they tend not to form a pointy protrusion into the white matter. And at that level, we don't call that a lateral horn, but we do recognize another collection of preganglionic visceral motor neurons. Only in the sacral cord, they're parasympathetic, whereas in the thoracic cord, they're sympathetic. All right, another key distinction that we need to make when we look at the spinal cord is in the region of the dorsal column. So remember, the dorsal column is what we find out here, with dorsal being shown to the top of the image and ventral or anterior to the bottom. What I want you to notice is that in the dorsal column, there is more white matter in the cervical region compared to the lumbosacral enlargement for reasons that I just illustrated for you. But I also want you to note that in the cervical cord, there is a distinction between a lateral component of this white matter and a medial component. And as we talk about our pathways for mechanosensation, what you'll discover is that the medial component is what we find at lower levels of the spinal cord. But as we get into the region of the cervical enlargement, we begin to add white matter from the sensory inputs that are attached to that cervical enlargement, serving the upper extremities. Well, that white matter begins to add up in the lateral aspect of this dorsal column, in the upper thoracic cord, and then we really see it adding up as we move through the cervical enlargement. So look for those divisions of the dorsal column when we look at cross-sections through the spinal cord. And we'll give these divisions names. I don't need to tell them to you now, but just note that in the cervical cord, the dorsal column is subdivided, whereas in the lumbar and sacral levels, there's no such subdivision of the dorsal column. Okay, now I think we're ready to name some of these details and give you a chart that will allow you to explore on your own cross-sections through the human spinal cord. So this is a chart that lays out for you some important internal features in terms of gray matter and white matter. And for white matter, I'm giving you the names of some of these important tracks or pathways that we will talk about in detail when we talk about mechanosensation and our systems for pain and temperature. Well, let me orient you to this chart. We have a list of internal features of the spinal cord in columns, and then our spinal cord segments organized according to cervical, thoracic, lumbar, sacral, and the one coccygeal segment. And what I've provided for you is a check mark to indicate that that gray matter structure is present. So as you've just seen, in an array of cross sections, the dorsal horn is present in each level of the spinal cord, as is the ventral horn. Okay? So we have a check mark for each of these longitudinal divisions of the spinal cord. The lateral horn is that special feature indicative of the preganglionic sympathetic neurons, and that's only found in the thoracic cord. So please note that. The lateral horn is a feature of the thoracic cord. Now, the rest of this chart pertains to white matter. So just with respect to the overall abundance of white matter, as I've suggested, there is less and less white matter as we proceed from superior to inferior. And with respect to the individual pathways or the individual tracks of white matter that will be important for mechanosensation and pain and temperature sensitivity, as well as for motor control, we also see a general superior to inferior progression from more white matter superior to less white matter on the inferior side. So these terms here, gracile tract, cuneate tract, these are the pathways that we find in the dorsal column. The gracile tract pertains to the lower extremity, the cuneate to the upper extremity. So the gracile tract begins to build up in its complement as we pass through the portion of the spinal cord that receives input from the lower extremities. We don't have a cuneate tract until we get into the upper part of the thoracic cord. And then once we do, we rapidly add axons that are adding somatic sensory signals to that dorsal column. Now, I'm going to jump to the far right column because this also is a somatic sensory pathway. This pathway, called the anterolateral system, is found, as its name suggests, in the anterior and lateral white matter of the spinal cord. And this is a system that is conveying pain and temperature signals from the body up into the brainstem, into the forebrain. So here we have a progressive addition of white matter as we go from inferior to superior levels. Now I'm talking about an ascending sensory pathway, which is why my arrow is going up. Now finally, we have two pathways that pertain to motor control identified here. A lateral cortical spinal tract, which is really the major motor control pathway that I will emphasize for you in this course. But there's also a smaller, minor ventral cortical spinal tract. The major pathway, the lateral cortical spinal tract, descends with a progressive termination of axons as we go from the upper part of the cord to the lower part of the cord. And really, much of this pathway is concerned with what we do with our arms and our hands, and to perhaps a lesser extent, what we do with our legs and our feet. This pathway is all about controlling voluntary movements, which mainly involves our arms and our legs. So there's not as dense a projection to the thoracic cord, but we still see the pathways that are working their way down to the lumbar segment. And then once we get below the lumbosacral enlargement, this pathway really drops out of the spinal cord. Now, this minor pathway, the ventral cortical spinal tract, it's a relatively small contribution, and the axons tend to project along the length of the spinal cord, perhaps with some bias towards the lumbar levels. All right, well, now that we've had a broad conceptual overview of the spinal cord, it's time to actually look at cross-sections and see these features directly. So I would ask that if you have Sylvius software, go ahead and open it and follow along with me. If you don't have the software, that's fine. You can just continue to track with me as we shift now to Sylvius and look through some representative cross-sections of the spinal cord. Okay, so I'm going to open up the Brainstem Cross-Sectional Atlas in Sylvius, and I'm going to click on All Structures. And so this opens up a view of histological cross-sections through the brainstem that extend all the way into the spinal cord. So in a different tutorial, I've taken you through the brainstem. So what I'm going to do is jump right to the spinal cord. So I can do so with my navigational window to the left, either just pulling on the sliders or selecting a thumbnail of interest. So what we have here is a cross-section through the cervical segments of the spinal cord, and we can appreciate that from our navigational window. But now I also would expect you to recognize that based on the large ventral horns that we see, plus the abundance of white matter that we see around the outside of this gray matter. And if one looked specifically at the dorsal columns, you'll note that there is a subdivision. And of course, with the color code, we make that easier for you to appreciate. But even without the color code, with a careful eye, one can denote the presence of a small septum that divides the dorsal column region into two parts, a medial part and a lateral part. All right, and just a few other features of note. We've already talked about the very large ventral horns. In the dorsal horn, we have a very striking region of the dorsal horn that corresponds to Rexsid's lamina number 2, and that's a region called the substantia gelatinosa. And that's this very bright region here in this section that's made to look like a myelin stain. It's bright because there is a compact zone of small cells with lots of synaptic inputs and not much myelin in this region of the dorsal horn. Now, in the surrounding white matter, I'd point out a few prominent features. I mentioned one major descending pathway. This is the lateral cortical spinal tract. So this is a major means by which the motor cortex sends motor command signals down to the circuits of motor neurons in the spinal cord. So I'm just going to make sure this is selected and just allow you to see through the thumbnails. And so this allows you to note how the cortical spinal tract gets smaller and smaller and smaller as we go from superior to inferior levels of the spinal cord. Okay, back up to the cervical enlargement, and we're ready to move on now to look at the thoracic cord. So think about what's going to be the difference between the thoracic cord and the cervical cord that's in front of us right now. Well, hopefully you have in mind a couple of things. Maybe a small reduction in the amount of white matter that we would see, but perhaps a large reduction in the size of the ventral horn, because we know that the thoracic cord doesn't have to supply motor neurons that govern an appendage, so we would expect a smaller ventral horn there. But you should also now think about the lateral horn, which is a distinctive feature of the thoracic cord. So it's sectioned down one level, and sure enough, we have less gray matter in the ventral horn, and we have a prominent lateral horn, which is mainly there because of a column of cells that are preganglionic sympathetic neurons. That column is called the intermediolateral cell column, and it is essentially why there is a lateral horn in the thoracic cord. We still have a dorsal horn, we still have a beautiful substantia gelatinosa, and we still have quite a bit of white matter because we're still relatively high in the spinal cord at this level. But as we get down to the lumbar enlargement, now the relative proportions of white matter to gray matter seem to be quite different. There's less white matter in absolute terms, but there really is a dramatic difference in the apparent ratio of white matter to gray matter. That's because we're losing white matter, but we also are now adding a very large ventral horn that accommodates the muscle mass of the lower extremities. So we have a prominent ventral horn. Of course, we would expect a sizable dorsal horn receiving sensory signals from the mass of our lower extremities. And then in the dorsal column, I would just highlight for you the fact that we don't have a subdivision that we saw in the cervical and even the upper thoracic cords in the dorsal column, but just a single dorsal column system, which is called the gracile tract. So if I select that, and now we can work our way back up the spinal cord, you'll see how the gracile tract is present in the more medial aspects of the dorsal horn, whereas this lateral region is called the cuneate tract. The cuneate tract basically accounts for the addition of sensory axons from the upper extremities. All right, so returning to the lumbar sacral enlargement, and now we can progress to our most inferior section, which is from the sacral cord. So now again a even greater disparity between the relative amounts of white matter and gray matter. We still have a large ventral horn because we're still in the region of the lumbosacral enlargement serving the lower extremities. We still have a prominent dorsal horn, but we have much less white matter. That's especially evident in a smaller lateral cortical spinal tract that we have in this region, as we're nearly below now the level of gray matter that is going to be subject to the control of our volition. That's one way to think about this lateral cortical spinal tract, that it represents the motor cortex's command capacity over the output of the spinal cord. Well, I think that will do it for this tutorial. I hope this has given you a helpful means of orienting your conceptual knowledge about the organization of the spinal cord to the actual appearance of this beautiful structure in histological sections. What we have in front of us is a hemisphere that has been sectioned in the mid-sagittal plane. And the purpose of this lesson is to focus on the spaces within the brain. You'll recall that all of this beautiful brain formed from the walls of a tube. And the lumen of that tube is still present in the adult form of the brain. It's what we call the ventricular system. And it's in the ventricles that cerebral spinal fluid is produced. And we want to observe those ventricles and follow the flow of cerebral spinal fluid from the ventricles to the space outside of the brain, and then finally back into the bloodstream. So let's begin by looking at the largest of the ventricles. It's called the lateral ventricle, and it's found just on the other side of this membrane. So what I'd like to do is I'd like to gently dissect away that membrane so that you can see the lateral ventricle. And I can do that by just cutting into this space just underneath the corpus callosum. And I'm going to cut a bit of a flap so you can see that there really is a space on the other side of this membrane. So now with that opening made in the septum pellucidum, you can see a large space within the anterior part of the forebrain. So this is called the lateral ventricle, and it extends forward into the frontal lobe. It runs through the parietal lobe, and even extends back into the occipital lobe. And then there is a branch of this space that wraps around into the temporal lobe on the lateral side of the brain. So the lateral ventricle is by far the largest of these ventricular spaces in the brain. Now, much of the cerebral spinal fluid is produced in that lateral ventricle, and it passes from the space within the hemisphere to the third ventricle, which is found right here along the midline of the diencephalon. Now, in order for fluid to get from the lateral ventricle into the third ventricle, it has to pass through a narrow passageway called the foramen of Monro, or the interventricular foramen. And I'm going to pass this tube through that space to demonstrate that channel. There we go. So now you can see the tip of the tube that has passed through the foramen of Monro and allows the communication of cerebral spinal fluid from the lateral ventricle to the third ventricle. Now, notice that
the lateral ventricles don't directly communicate one to the other, that septum pellucidum that I dissected away is a barrier that keeps the fluid from one ventricle from getting to the next. So here we have our third ventricle, which again is a narrow slit that is bounded on either side by the hypothalamus and the thalamus above. And cerebral spinal fluid flows from this third ventricle down into the fourth ventricle through the smallest of the channels that we have within our brains. And that channel is a narrow space or a narrow aqueduct that runs through the dorsal aspect of the midbrain. And this thin tube that I have is just about the dimensions of that channel. And let's see if I can pass this tube through that channel from the third ventricle down into the fourth ventricle. And indeed, we see this tube emerging now in the fourth ventricle. In fact, the fourth ventricle allows for the escape of cerebral spinal fluid. Cerebral spinal fluid exits the inferior limit of the fourth ventricle, where there is a space between the cerebellum and the medulla. That space is called the foramen of Magendie. There's also another small aperture deep in the lateral recess of the fourth ventricle called the foramen of Luschka. So from that point, cerebral spinal fluid exits the ventricular system and flows around the entire cerebral hemisphere in the space underneath this tissue called the arachnoid. So the tissue that's surrounding the hemisphere here normally contains about 1 millimeter or 2 of cerebral spinal fluid that forms an important physical buffer around the outside of the brain. Finally, this tissue allows for the drainage of cerebral spinal fluid along the dorsal midline. These rough features of the arachnoid right along the dorsal midline here, these are called arachnoid granulations. And these are one-way valves that allow cerebral spinal fluid to leave the subarachnoid space and drain into the superior sagittal sinus and then back into the venous blood. Now, before we conclude this lesson, and while we have this brain in front of us, I want to highlight one somewhat unusual feature of this particular specimen, and it has to do with the cerebellum. Now, I want to show you the cerebellum from what is likely a normal brain in order to compare with this specimen that we have in front of us. So here we have a cerebellum from an individual who died without neurological complication. And I want you to notice how tight and full are these tiny little branches or folia that form the cortex of the cerebellum. In contrast, this other brain has an unusual amount of space between the folia of the cerebellum. These spaces are indicative of one particular pathology that seems to result in the atrophy of this anterior and medial part of the cerebellum, and that condition is chronic alcohol abuse. So although I do not know these individuals from whom these brains were obtained, nor do I know their medical history, the appearance of the cerebellum in this specimen is highly indicative of chronic alcoholism compared to what we see in a healthy-looking cerebellum. And this alcohol abuse that I suspect might have afflicted this particular brain is likely to have impaired the coordination of gait and other aspects of locomotion in this individual in life.
Hello everyone. Welcome to this tutorial on the anatomy of the internal capsule and the deep gray matter. Our principal core concept for this session is to simply, again, try to appreciate the complexity of the human brain, but to do so by guiding you through some principles that will help you understand that complexity. My learning objective for you today is that I want you to be able to describe and even to sketch the relations of the deep gray matter structures that we find within the core of the forebrain with respect to the internal capsule. And this is an important white matter structure that runs right through the middle of each hemisphere. And I want you to appreciate the relationship of the deep gray matter to the internal capsule as viewed in both the coronal and the axial planes of section through the forebrain. What I want you to confront is the internal anatomy of the forebrain, what we find deep in the center of each hemisphere. And the chart here on this slide really provides you with a summary of what I want you to apprehend. So, from an embryological standpoint, the cerebral hemisphere consists of the telencephalon, which is the largest component of the cerebral hemispheres, and then just inferior and a bit caudal, we have the diencephalon. Now, what I want you to be able to do is to consider these various derivatives of the embryological telencephalon and diencephalon. And mainly what we're going to focus on is one bit of white matter called the internal capsule, and the relationship then of these gray matter structures relative to the different components of that internal capsule. Okay, so yes, we're going to see the cerebral cortex and briefly the hippocampus and the amygdala and the temporal lobe, but really our focus is going to be here in the basal ganglia and in the diencephalon, and in particular in the thalamus. So what I want you to be able to do is to recognize how is the caudate nucleus, the putamen, the nucleus accumbens, and the globus pallidus positioned relative to the internal capsule. And then how does the thalamus and the hypothalamus fit in? And essentially the answers to these questions will be either medial or lateral to the internal capsule. And then to complete the picture, we can consider the placement of some additional white matter structures, such as our commissures of the forebrain, the corpus callosum, and the anterior commissure. We'll see how the fornix fits in. And then I want you to appreciate how the ventricles that are associated with the forebrain, the lateral ventricle and the third ventricle, are arranged relative to the deep gray matter in the internal capsule. Okay, well, let me show you what I'm talking about. If we look at the lateral view of the cerebral hemisphere, and if it were possible to look right through the cortex, as we're doing here in the illustration to the upper right, what we would see are a series of gray matter structures arranged around white matter. And the gray matter now are isolated for you on the left. And the colors that we see here are indicative of some of the major divisions of this deep gray matter. This purplish color shows us the basal ganglia. And the basal ganglia that can be seen from the lateral view includes the caudate nucleus, the putamen, and the nucleus accumbens. Now you'll notice that they really don't look like they're separate structures, but rather just a complex, three-dimensional, unitary structure. And indeed, that's a very helpful way to think about the basal ganglia. Together, these three structures are called the striatum because of the arrangement of cells in this general region of this structure between the putamen and the caudate divisions. Well, I want you to understand how is that striatum, or these divisions of the basal ganglia, arranged relative to the thalamus and to the internal capsule. And you see the thalamus is positioned slightly posterior, but on the medial side of the putamen. Hopefully you get a sense of that. But there is this internal capsule that I've been referring to. And let me just sort of illustrate that for you here so you know how that fits in. The internal capsule is this massive system of white matter that runs between the cerebral cortex and everything that falls below the cerebral cortex. So that's going to include the thalamus, it's going to include the brainstem, and it's going to include the spinal cord. Now, many of these axons, as I've been illustrating here, are heading in the descending direction. Some of them will actually go all the way down into the spinal cord. Other axons, namely those that are coming from the thalamus, are actually projecting back to the cerebral cortex. And so what we have in each hemisphere is this massive fan of white matter that comes together in a compact bundle and passes between parts of the basal ganglia and the diencephalon as this white matter courses from the forebrain down into the hindbrain. Well, if we turn now to our enlarged view of the basal ganglia, this is exactly where we would see those axons. They would be slipping in this little cleft here between the body of the caudate nucleus, the thalamus, and the basal ganglia, the putamen, the nucleus accumbens. Now, there's another component of the basal ganglia that we don't see here that's also with the putamen on the lateral side of this internal capsule. And that structure is the globus pallidus. The globus pallidus would sit right about in this region here, completely enveloped by the striatum. Now, if this seems a bit messy, I suppose it is, but what I really want you to appreciate is the way that these axons of the internal capsule provide such a useful landmark for us neuroanatomically, because they help us understand where you would expect to find one of these important gray matter structures. And that's really what I hope for you to get out of this tutorial. Well, let's back up a slide and I'll tell you exactly where you should expect to find these gray matter structures relative to the internal capsule. Okay, so again, our focus is primarily on the basal ganglia and the thalamus. What we would expect to find are two structures that are found on the medial side of the internal capsule. Those would be the thalamus and the caudate nucleus. On the lateral side of the internal capsule, we would expect to find the putamen and the globus pallidus. Now, what about the nucleus accumbens, you might ask? Where would we find that? Well, the nucleus accumbens is really neither medial nor lateral. I would say it's actually inferior to the internal capsule. And you'll see what I mean when we look through sections of the brain. Well, let's do that now. And I'd like to segue to a segment that I recorded in the Brain Anatomy Lab. And you'll see me directing your attention to these various components of the deep forebrain. So take some time, take that in, refer to the tutorial notes that I've given you, and then after we go to the lab, we'll conclude by looking at these sections in Sylvius one last time. Okay? Well, I'll see you shortly in the lab.
What we have in front of us is a coronal slab through roughly the center of the forebrain. And I want us to examine the features that we find deep within the brain. On the outer surface, we have the cerebral cortex, which is just a few millimeters of gray matter thick. And then beneath the cerebral cortex, we have white matter. And that white matter runs in various directions throughout the hemisphere. There are some special white matter structures, such as the corpus callosum, that are obvious in this section. And then there's one that's not quite so obvious, but it's worth pointing out, and it's called the internal capsule. The internal capsule is a band of white matter that allows the cerebral cortex to send axons to the diencephalon and the brainstem and spinal cord below it, as well as for structures like the thalamus and certain cell groups in the brainstem to send axons up to the cerebral cortex. For our purposes here, the internal capsule is a landmark that helps us orient the rest of the deep gray matter visible in this section. On the medial side of this deep gray matter, we have a structure called the caudate nucleus. And the caudate nucleus is part of the basal ganglia. The caudate nucleus sits just medially to the internal capsule, and it forms the lateral wall of the lateral ventricle. Now, just on the lateral side of the internal capsule is another bit of gray matter that's very similar to the caudate. Its name is the putamen. So the putamen is this bit of gray matter that sits out here lateral to the internal capsule. Now there's a triangular region between the putamen and the internal capsule that is yet another deep gray matter structure, a different part of the basal ganglia that we call the globus pallidus. There are actually two parts to the globus pallidus. This pin is currently in the internal segment of the globus pallidus, and just on the outer side of that pin is what we call the external segment. Now, these three deep gray matter structures are principal components of the basal ganglia, and it's important to know their relations one to the another and to the internal capsule. Also in this view, not in the basal ganglia, but down here in the anterior part of the temporal lobe is the amygdala. The amygdala is a large mass of gray matter found in the anterior medial temporal lobe just in front of the hippocampus. And the amygdala is very much involved in alerting the rest of the brain to situations that might potentially be threatening or otherwise alarming. Meanwhile, these structures that we've pinned here, the basal ganglia surrounding the internal capsule, these are structures that are all about movement. So now what we have in front of us is another coronal slab taken just about a centimeter in front of the first slab that we were looking at. And again, I want to highlight the internal capsule, which is this band of white matter that helps us orient ourselves relative to the gray matter that surrounds it. And on the medial side of the internal capsule is the caudate nucleus, forming the lateral wall of the lateral ventricle. And on the lateral aspect of the internal capsule is the putamen. Now, in this slice, we don't have the globus pallidus because the globus pallidus is a structure that we only see near the middle of the basal ganglia. We're just in the front part of the basal ganglia, and it's worth focusing in this region so that we can highlight a portion of the basal ganglia where the putamen and the caudate nucleus join together just inferior to the internal capsule. This part of the basal ganglia we call the nucleus accumbens, and I've given it a flag with the letter A for nucleus accumbens. Each of these three parts of the basal ganglia, the nucleus accumbens, the putamen, and the caudate, they're all about helping to regulate movement. The putamen is involved with regulating movement of the body, right here. The caudate nucleus is concerned with movements of the eyes, and with movements of the eyes go movements of the mind and of thought. So the caudate nucleus is involved in modulating movements of thought. Down below, the nucleus accumbens is involved with modulating movements of emotion or mood. So we can think of these components of the basal ganglia as all being involved with the modulation of movement. What distinguishes them are their anatomical connections and also the functions upon which they operate. Now this coronal slab in front of us is about 2 centimeters posterior to the last one we just saw. And I want to draw your attention into the anterior and medial part of the temporal lobe. Where 2 centimeters in front we had the amygdala, now we have a structure known as the hippocampus. And the hippocampus is one of those fantastic names in brain anatomy that tell you something about its shape. The hippocampus is so named because of the curvature of the cortex, as this structure folds in upon itself, much like the body of the seahorse. Hippocampus is a Greek word for seahorse. And I think you can appreciate the curving nature of this medial margin of the temporal lobe forming the hippocampus. We see it nicely on both sides, forming the medial edge of the temporal lobe, and also forming the medial wall of a bit of the temporal horn of the lateral ventricle. So the hippocampus protrudes towards the lateral ventricle from the medial edge. Now what we have in front of us is an axial slab from a different brain, and we want to look again and find the same set of structures, the internal capsule, components of the basal ganglia, and the hippocampus. Now let's begin with the internal capsule. From this perspective, the internal capsule makes a bit of a chevron shape in the brain. We can speak of an anterior limb to the internal capsule and a posterior limb of the internal capsule. Now let's look at the anterior limb of the internal capsule. We can identify it again with a little flag, just to hold your attention on this bit of white matter. On the medial side of that internal capsule, we have the caudate nucleus, which, as you can see in this plane, forms the lateral wall of the lateral ventricle. So this space we see here, this is the lateral ventricle, and note how the caudate nucleus is protruding out into it. On the lateral aspect of the internal capsule, we have the putamen. So there is our putamen. And in this specimen, you can probably notice there are multiple little markings here.
And these spots probably reflect the consequence of chronic hypertension. The small black dots you see are blood vessels cut in cross-section, and the whitish spots around them are probably some scar tissue that has been laid down by activated astrocytes. And these spots represent the consequence of having tiny blood vessels pulsate under elevated mean arterial pressures for an extended period of time. And you can think of these as local regions of microtrauma deep in the tissue.
Now, just medial to the putamen, but still lateral to the internal capsule, we have the globus pallidus. So there's our globus pallidus in this specimen, with the boundary between the putamen and the globus pallidus being visible right along this margin right there. So we have putamen lateral to that margin, and then globus pallidus on the medial side. Now also in this tissue section, we have a nice look at the hippocampus. So here again is the hippocampus, which is on the medial edge of the temporal lobe as this cortex wraps around itself into the shape like the body of a seahorse.
Okay, well now that you've actually seen the human brain in the lab, let's look at some brain sections through Sylvius just to recap the structures that we've been viewing. So what I've done is I've entered Sylvius and selected sectional anatomy and photographic atlas. And I think what we'll do is we'll start in the coronal plane. So I'm going to simply tap on the coronal plane with a selection of our thumbnail image. And let's now just walk through the brain in the coronal plane. And I've selected the filter set that will colorize components of the deep gray matter as we encounter them. So let's begin to section about a few centimeters from the anterior pole of the hemisphere. And I just want you to notice that there's no deep gray matter a few centimeters into the brain. Just a reminder that the basal ganglia are set in towards the center of each hemisphere. So if we section back now another centimeter or two, now we begin to see the basal ganglia as they're arranged around the very anterior edge of the internal capsule. We call this the anterior limb of the internal capsule. Now notice what we see. In the red coloration, we have the caudate nucleus, which is on the medial side of the internal capsule. And notice how the caudate nucleus bulges outward towards the lateral ventricle. It forms the lateral wall of the body of the lateral ventricle. Now, on the lateral side of the internal capsule, we have the putamen, And again, notice it's lateral to the internal capsule, which is just starting to make its appearance here. And again, we call this the anterior limb of the internal capsule, as these axons are streaming from mainly the cortex that is in the front part of the frontal lobe. Now, not labeled here in Sylvius, although we certainly could have here, would be the nucleus accumbens. So let me just highlight that region of the nucleus accumbens. It's basically the ventral part of the striatum where the caudate nucleus and the putamen wrap around and come together just inferior to this anterior limb of the internal capsule. So this is roughly where we would find our nucleus accumbens. Now that's one of those nice terms in neuroanatomy that sort of tells you what it means. This nucleus accumbens is recumbent or reclining upon the septum, which is this medial structure that divides the walls of the lateral ventricle and the inferior part of the posterior frontal lobe. So the nucleus accumbens is the nucleus that's lying down on the septum. Okay, let's go back a section. And at some point, we expect to find the globus pallidus, do we not? And indeed, we see it here. So the globus pallidus is found in green, and it, like the putamen, is lateral to these axons of the internal capsule. So here's the internal capsule. So again, we have globus pallidus and putamen that are found lateral to the internal capsule. And on the medial side, we have the caudate nucleus. Now, if I can draw your attention towards the center of the section, you'll see deep into the lateral ventricles, there's some gray matter that seems to be sticking out at us. I'm referring to these two bulges right there and right there. Well, hopefully you might have in your mind that perhaps that's the thalamus. It's medial, it's on the medial side of the internal capsule, it's a little bit posterior, a little bit set back from the large part of the basal ganglia, and indeed that's going to be the thalamus that we will see as we cut through into our next section. So now what we find is all the three major components of the basal ganglia that we saw in the last section: the caudate nucleus on the medial side of the internal capsule, the putamen getting smaller but it's still present, and the globus pallidus also present but getting smaller. And just to be more clear, here's our internal capsule. Now we are getting from the anterior limb into what we call the posterior limb of the internal capsule. And one hallmark of getting into the posterior limb of the internal capsule is the development of the thalamus on the medial side of the internal capsule, just inferior and medial to the caudate nucleus. So this is the thalamus. The region just inferior and anterior to the thalamus is where we would find the hypothalamus. Hypo suggesting below or inferior to the thalamus, and indeed that's where we find it. You may be wondering about the structure that is colorized yellow here. This is the amygdala. The amygdala is sometimes considered part of the basal ganglia, but I think it's probably most helpful to you not to consider it part of the basal ganglia, even though it's included here in Sylvius under our basal ganglia module for historical reasons. But as we'll get to talk more about the amygdala, you'll see that its functions can largely be differentiated from those that pertain to the basal ganglia. Although for the aficionados out there who may be studying the basal ganglia and the amygdala, you know that from a different perspective, they do indeed have many embryological, histological, and physiological features in common. Well, that's a subject for a more detailed course at some other time. But for our purposes, let's consider the amygdala separate from the rest of the basal ganglia. Well, just for completeness, we'll go back one more section, and what we'll see now is that we are behind now, or posterior to the putamen, the globus pallidus. We still have a little bit of caudate nucleus, right? Because the caudate nucleus is curving around, and we actually see some of it in the temporal lobe, just on the dorsal margin of the temporal horn of the lateral ventricle. And that's because this caudate nucleus forms this C-shaped structure with a long tail that extends into the temporal lobe. So the cut that we've made shows the caudate nucleus in 2 locations within each hemisphere. All right, now let's go through the same experience, only now in the axial plane. So let's section down from above, and if we enter the hemisphere a few centimeters below the dorsal aspect of the hemisphere, we are clear above the basal ganglia. So what we see is simply cortex and the white matter at the center of the hemisphere. If we go in one more section, now I think it's very clear that the caudate nucleus is on the medial side of the internal capsule. So here's the caudate nucleus, and what we see is mainly the posterior head and the body of the caudate nucleus. Once again, we see the thalamus peeking out at us from the medial side of the internal capsule, but set back into the lateral ventricle. You may be wondering where the internal capsule is on this view. The internal capsule is going to come through this white matter in this region, just on the lateral edge of the caudate nucleus. Okay, so let's go down one more section. And now we see our 3 major components of the basal ganglia that we've been featuring in this part of the tutorial. We have the head of the caudate nucleus and a little bit of the tail. We have the putamen, which is shown here in blue, and we have the globus pallidus, which is colorized here in green. Now the thalamus is located in this region right back there. Okay, now I realize it's a bit difficult to appreciate the internal capsule here, but this is an important view of the internal capsule. It's running between the head of the caudate nucleus and the putamen and the globus pallidus, and it's also separating the globus pallidus, the putamen, and the thalamus. And you'll notice how I'm highlighting this internal capsule here. I'm doing it in a way that hopefully brings to light the posterior and the anterior limbs of the internal capsule. So this is the posterior limb of the internal capsule, and this would be the anterior limb of the internal capsule. And you may also notice there's a little wisp of white matter that's running right between the 2 hemispheres in that location. That has nothing to do with the internal capsule. That's actually at a more inferior position. This is the anterior commissure, one of those great commissures that interconnects gray matter structures in the cerebral cortex. Okay, so I want you to appreciate now the layout of this deep gray matter relative to this internal capsule, because in this one axial slice we see all the important relations that I want you to understand. The globus pallidus and then the putamen are lateral to the capsule, and the caudate nucleus and the thalamus are medial, with the caudate nucleus on the medial side of the anterior limb of the capsule and the thalamus on the medial side of the posterior limb of the internal capsule. Okay, well, I hope you will have a chance to exercise your knowledge of this deep gray matter. One way that I would recommend that you do it is now on your own, if you have a copy of Sylvius, enter our MRI atlas. This is a T1 weighted MRI atlas of the human brain. And rather than colorizing the brain, we've placed these small little dots on the brain. And if you select a dot, you will see opportunity to identify the structure that's labeled. If you click on that structure, now there's information available in a text field over here to the right. So what I would invite you to do if you have Sylvius, is to section through the brain. And since this is a MR study of a human brain, we have a much greater density of images, and I think that will be of benefit to you. And I hope you can recognize in this T1 weighted image many of the structures that we saw in the lab in a fresh human brain and also in Sylvius. And what I would invite you to do is to walk through and make sure you can recognize the structures, not based on how well you can interpret an MR scan, but rather based on your conceptual knowledge of where you expect to find deep gray matter relative to the internal capsule and the other important features of the interior of the hemisphere. Okay, well, with a little practice, I'm quite confident that you can acquire these competencies, and you will very quickly be navigating your way through sections of the human brain.